Okay, hello everyone. We're just going to give it 1 or 2 minutes and then we're going to start today's webinar. Okay, let's wait 1 more minute and then we start today. Okay, let's get started. So welcome everyone to this session of the Microbiome Informatics webinar series. And today I will introduce a genome-based taxonomy, phylogenomics, and we will focus on an archaeal case study. Before we dive into it, I want to quickly introduce myself. So I'm Chris Rinke. I am a senior lecturer and future fellow and group leader at the Australian Centre of Ecogenomics at the University of Brisbane here in Australia. And my group focuses on microbial ecology and microbial genomics. Main topics are ocean and river microbiome. We do work with microbial dark matter, exploring of new lineages. And recently we have shifted more and more to microbial plastic degradation because we are in a plastic waste crisis, and microbes have some very interesting enzymes that could help us to degrade plastic waste and to tackle that crisis. Also, in case you're wondering where Brisbane is located, it's in the north of Australia. You can see here, if we go over here, that is Australia. We have Sydney down here with the famous Sydney Opera House. And then up here in green, that is the state of Queensland with Brisbane. And if you zoom in a bit, in the northern part of the state, that's Cairns. That's where we have the Great Barrier Reef. And then in the south here, that is Brisbane. Here's the main campus of the University of Queensland. And many people think that Australia is mostly dry desert, the outback, and we have that inland. But along the coast, we have I would say one of the most beautiful beaches in the world, probably. I'm biased. We also have gigantic sand islands like this one. That's Lake McKinsey on Fraser Island. It's a freshwater lake on a sand island, and most of it in the north of Queensland it's tropical, and in the south here around Brisbane it's subtropical. So we have a lot of tropical and subtropical rainforests. Okay, that is Brisbane and Queensland. But now back to the topic at hand today. A genome-based taxonomy, phylogenomics, and an archaeal case study. So what I would like to do today is first, we're going to do a deep dive, not too deep, right, into nomenclature and taxonomy to dissect those two terms so we have a clear understanding what they mean, what the difference is. Then we will quickly look into the species concept, the prokaryotic species concept and the definition. And then finally, we look into a genome-based taxonomy, and that I will do that with the Genome Taxonomy Database, the GDB, since I'm a member of the curation team of GDB, and we'll apply an archaeal case study to show you how we standardize taxonomy. Okay, let's dive into it. This slide is an overview that show you that nomenclature and taxonomy, it's not a static concept, it's actually evolving over the years, right? If you look here on the left, let's start in the early 2000s. For the nomenclature, we do have the prokaryotic codes. And then as taxonomy resource, we have NCBI. And the way NCBI works, it's actually a collection of taxonomic opinions. So if someone proposes a new lineage, publishes that, that's submitted to NCBI, and NCBI incorporates that, right? So there's no overall authority or system that checks those submissions in terms of, you know, what ranks there are. No, it's basically a collection of taxonomic opinions submitted by the user. Now, in the early 2000s, we did have rather high costs to sequence and assemble isolate genomes, right? So there were just a few. And yes, that was the start of MAGs, of metagenome assembled genomes, that could be assembled at the beginning from low complexity environment, like acid mine drainage, right? And that was a very labor intensive manual process in terms of the bioinformatics. Now, that changed over the years. If we switch to, excuse me, to 2010 here, some people also call this the start of the genome area. Main reason for that being is that we have a large number of metagenome assembled genome MAGs that became available, right? Mainly because the costs for sequencing and also the assembly time, meaning that decreased quite significantly, and that allowed studies to come up with like large sets of MAGs in this case. Still, NCBI taxonomy was the taxonomic resource. Now that changed a bit more recently, 2017 to 2019, when GDB was created, a genome taxonomy database, and that was mainly to accommodate this large number, because at this point, you know, the MAGs were exploding. There were studies with MAGs. I was involved in one as well, where we found like 8,000 new genomes, and that was quickly superseded by another study with like 20, 50, and I guess now they have studies with like 70, 80,000 genomes, MAGs in one study. So we needed a system to accommodate this large number of new genomes, and that was the start of the GDB. We do have then at this point two taxonomic frameworks, and they don't necessarily agree with each other, right? And I will go into that in a little bit more detail. Then fast forward to today, 2022, we have still two main taxonomic frameworks, and we do have now a new code of nomenclature, that is the C code. I should say that is actually still being actively developed, right? So probably will still take a little bit more, maybe next year it will then roll out. But yeah, that's how it stands right now. And we will talk about GDP a bit more, and I'll also have a slide about the C code towards the end of my presentation. Now, let's look into some terms we need for today's lecture. So first, a taxon. What's a taxon? You can see it's defined as a group of organisms, and the plural is taxa, right? So I think everybody's familiar with that. Now, how do we organize taxa? We use the convention of Linnaeus, and that was proposed already quite a while ago in 1735. Maybe the theme here is, if it ain't broke, don't fix it, right? It works very well. We're still using it, not only for prokaryotes, but for all living things on Earth. Cellular living things on Earth, I should say, right? So what's in there? Taxa organized into hierarchy. We can see this here, right? It goes from species, genus, family, order, class, phylum, all the way to domain. And the other part of that is that we have a binomial nomenclature, meaning that species are binomials. For example, species name Posidonia alphae, right? Has like two parts, this species name. There's the species part, and then the genus part is in there as well. So that's a convention, I guess we can, or everybody agrees on, which is obviously very good. Then taxa, right, should also reflect evolutionary relationships. As seen here, that is Darwin's first tree, right, when he started. So that should just illustrate that if we talk about a taxa, we want it to be a monophyletic group, right? Ideally, that's what it should be. Okay, now one thing we have to clarify, and I think that's probably one of the take-home messages of this webinar today, is the difference between nomenclature and taxonomy, because that has caused quite some confusion. Let's have a look at that. So in a nutshell, right, nomenclature covers how do we propose and assign names to taxa. It's really the naming of taxa. Versus a taxonomy, right? That is about how do we assign organisms to taxa, right? And how to provide taxonomic ranks. Okay, so what does that mean? Let's look at an illustration of those concepts. So let's say you have like strains, right? Or genome sequences of strains, right? Then the taxonomy is basically which box represents a species, right? So do you say, are those 5 strains a species? Are those 7 a species? Are those 9, right? At some point, we have to basically draw a line and say, okay, that represents a species. And that is what taxonomy does. Now, nomenclature is different, right? That's actually how do we call the box? Once we, and they have to work with each other, right? But we can actually call the box before we decide what the borders are, right? Or we can call the box and then change the borders later. So those are two things that are quite different, but they definitely interact with each other. I hope that makes sense to clarify those two terms for you. Now let's have a look into the nomenclature. What are the basics here that we need to cover? So you might be familiar with that, but the prokaryotic names, right? They are regulated by the International Code of Nomenclature of Prokaryotes, or ICNP for short, also known as the code. And that is the prokaryotic code I showed you in the slide earlier. Now, if there are new names proposed, right, they are reviewed by a committee, and that committee is the International Committee on Systematics of Prokaryotes. Abbreviation is ICSP here. And this committee reviews those new names and decides then if they work with the rules of the code and if they can be included, right? And I do have here an URL if you want to learn more about the prokaryotic code and the committee. Now, let's have a look at the names, names and type material, and how does the code work? I have this example here. So if you focus on this part in the middle here, so what do I mean by type material under the code? Basically, the way the code is organized, right, is that everything goes back to a type material. So let's start with the species here, right? Each species has a type, which is a type strain, right? That is the type strain for a species. If you have a genus, right, then the genus name obviously comes from a species. If you have a family name, then the family name is always derived from the type genus. If you have an order, then the order name is also derived from the genus. If you have a class, then the type is not derived from a genus, but from an order. And that is just how it evolved historically, right? There's no apparent reason that I know that the class is derived from the order. That's just what historically developed, and that's the system that's in use right now. Then if you go all the way up to the phylum, that phylum is the phylum name is derived from a type genus, and you can see added here that the rank of phylum was only accepted recently into the code in 2020. And before then, phyla existed, right? They were used in the literature, but the prokaryotic codes did only include the rank up to class, right? Not phyla, and that was changed recently. Okay, let's look at one example here then. That is Bacillus subtilis, right? So we know in this case Bacillus subtilis ATCC6051. That is our type strain. So then we know what is the species and also what is the genus, right? If we then want to name the family, we have to actually see what is the genus name and what is the stem of the genus name, and that really depends on many different factors. I'm not going to go into that, but all those names are Latin or Latinized, right? So in this case, the stem would be Bacil, and then we add the ending ACA, right, to say, to show that that is a family. The same for an order, right? We also go to the type genus. So that's again Bacille, and then the suffix for the order is ALIS. For class, in that case, the same thing. We go to the type here. That's the order, and then in this case, it's Bacilli, right, for the class. Okay, so far so good. I also added here a little summary that shows you what specific epithets are used for different ranks, and that goes from family, ACA, order, ALIS, AIA for class. There are some exceptions, right, to make it work with Latin, like here, and OTA is for phylum. There also are less common ranks, like subspecies and suborder, but they're not used very commonly, but they're also covered by the code, actually. Okay, let's look into, well, phylum names, because as I mentioned, that was changed in 2020. Well, maybe I should start here. So if you look in this example, right, the phylum name is derived from the type genus. That's now the convention we're going by. If you do this, right, so that's Bacille, and the ending is ota. So that would be Bacillota, right? But what is here? What do we have here? We have Firmicutes. And that was the phylum name that was used for, I would say decades, right? And that is also what's known in the literature. Now, why is it Firmicutes? Because under 2020, the code did not include the phylum, right? So the phylum name did not have to follow the rules. But now, since the rank of phylum is included in the code, it has to follow the rules. And what happened then is that the committee actually sat down and said, okay, how can we make those phylum names work with the new rules? That means that the name is based on the type genus. And that was then decided and published. And I have to say, not many people took notice until NCBI implemented those, right? And then there was a big uproar on Twitter. I copied like one thread here, where someone posted taxonomy drama popping off all over my feeds. And that was in December 21, when NCBI implemented those names. Here I should say, if you are interested in taxonomy, in nomenclature, or actually for most of the microbial genomics, microbial ecology, Twitter is really the best source, science Twitter. That's where I get the latest paper. You know what's going on, you learn about what's going on in the field. So I would recommend, if you don't have it, not associated with Twitter, but I think it's very helpful to have a Twitter account and follow what's going on there. Now, in this case, you can see what happened here in this meme is that according to the rule, right, that the phylum has to be derived from, the phylum name has to be derived from the genus, a few new names were introduced. In that case, exactly for Firmicutes, right, the new name is Bacillota, right? That caused quite some uproar and arguments, because some of those names, Firmicutes changed to Bacillota, Protobacteria based on the type genus are now Pseudomonadota, Actinobacteria now Actinomycetota, and Bacteroidetes are now Bacteroidota, right? Some of them are fairly easy to relate, like Bacteroidota. Others, like Bacillota and Pseudomonadota, are maybe a little bit more difficult. And people were arguing, no, we should actually keep the old names. We should have an exemption to the rules. And the other fraction argued that, well, we have rules for a reason, but we shouldn't start off with like putting in exemptions there. And my personal opinion, I was first also a little bit conflicted, and I kind of wanted to go with the old names. But the more you think about this, it makes sense to have a system in place that has rules and follow those rules, right? And it might take us a while to realize, to switch to Bacillota, but after a few years, I think that is done. We can always go back in literature and know that, prior to 21, this group was called Firmicutes. Yep. So another example that nomenclature in that case actually is evolving. Okay, let's have a closer look into the type material. So how do we actually, for example, decide? Let's say we have a family. How do we decide which genus is a type genus? And that goes back to priority and the date of priority. So in this case, we have again the family Bacilliaceae, right? And how do we know it's already named, obviously, but if it would not be named, how do we know what is the type material here, right? In that case, we know family, the name has to come from a type genus, right? Now we have many genera in this family. So what is then what is the type, right? In this case, it's the date of priority. So we really go by the genus that was described first. And in that case, the genus with priority is Bacillus, right? Because it was described first. And therefore, according to the code, yes, Bacilliaceae, right, will be the name for the family. We can do the same then for the order, obviously, right? You also go after the type, and then, for the class, you then go the type order. And if you have multiple orders, you would also go with the order that has the date of priority. So basically one that has been described first. So the good thing is there are actually fixed rules, right, for the naming of microbes or of prokaryotes in the code, which makes for more or less a stable system. Now, one more word about the type material, right? The type material here. Let's look at this example here. So let's go back to Bacillus subtilis again. And we now have an improved method in this example, right, to infer phylogenetic relationships. And what happens is, and that is a real example that really happened, is what we have known so far as Bacillus subtilis strains, right, appear to be or turn out to be polyphyletic. So the example you can see here, that is Bacillus subtilis, right? And you also have Bacillus subtilis here. And in the middle, you have a different species, Bacillus agri, right? So now the question is, what do we do with that, right? We need the type material. That's why it's really important. In that case, we know that is the type strain, right? The strain 6051. That is very helpful because that tells us, okay, the type strain is here. So that means that here is actually the species Bacillus subtilis, right? Because it has the type strain. That also means that those other, in that case, cultures that were named Bacillus subtilis are not Bacillus subtilis, and a new species name should be proposed for those, right? And this new species name then also has to have a new type strain, and that is then used for the species name, right? Okay, I hope that makes sense. Now, that is based on taxonomy. What if someone has a different taxonomic opinion, right? And that is totally fine, right? So, for example, an alternative opinion would be, okay, yes, I see what's going on here, but what one could do is say, well, why don't we just rename here Bacillus agri and say Bacillus agri needs to be reclassified as Bacillus subtilis, right? In that case, we would actually call this whole species Bacillus subtilis. Now, is that wrong or is it right? According to the code, it's neither, right? Because the code provides rules for naming of taxa, but it does not indicate how taxa should be circumscribed, right? The code does not care if you throw the bracket here for a species or throw the bracket here, right? So in a nutshell, the take-home message here is there is no official prokaryotic taxonomy, right? There's only a taxonomic opinion. So how do we solve this then? I will show in a bit. There is no official taxonomy, but there is like usually a common consensus that develops in the field, and that would be used then to deal with that case. Now, if you want to know more about type material, I would encourage you to go to LPSN, and in this case, that is the genus Bacillus. You can see it has the name when it was named. That's a genus. It talks about the etymology, right? Gender, 16S gene. It tells you what the type species is. So it's actually a very helpful website. I even think I highlighted this. Yes, you can see here that's the year of priority, right? If you have to decide if this is a type genus of a family, right? You have it here. You can go level down to the type species, right? And it's also important here that this name is validly published. So that means this name follows the code, has been verified, is validly published. In addition, I think it's interesting to look down here at the last part. those are synonyms, right? And that means that all those genera here at some point were believed to be different genera, right? But after the scenes have to be found to be actually included in the genus Bacillus. So that is good. If you find this in literature, you can actually go here and see, okay, that is a synonym for the genus Bacillus. Okay, but how do we name a species? That's probably an important aspect to cover as well. So if you have, let's say, a culture, and according to the code, you need a culture. Well, let's look into it. So if we have a culture, right, like a culture of a strain, then we designate a strain as a type, right, as a type strain. Random example here, Examplus demis, and usually a number MS23. What do we do to name this type strain? We have to submit it to a culture collection, and I show here the two largest one. That's ATCC in the US and DSMZ in Germany. Now there are many others that work as well, right? But it is according to the code, it is required to deposit the strain in 2 different culture collections, and that's really more a safety measure. Let's say something happens in one culture collection, right? You also have another one to come back to. Okay, so let's assume we deposited our strain into those 2 culture collections. Then we publish the new name, and obviously all information we have about the strain. In theory, we can publish that anywhere, right? It's easier though to publish it in the International Journal of Systematic and Evolutionary Microbiology, or IGSM short, because that is usually screened for novel names, right? So it makes it easier for the ICNP committee to actually find those new names, I would say, right? So once that is accepted and published, then your paper is effectively published. But there's more, right? Then the idea is to notify the IGSEM editors if they don't already know about this, and they will then put this new name on a validation list, right? And then that's a lot of work on the side of the editors, the committee, and they will actually go through and then see if those names correspond and align with the rules of the code, right? The ICNP. If they do, then they end up in the list that's going to be published, and then you have a validly published name, right? And that's basically mean it's included in the code. It is a valid name. Okay, I think that summarizes the nomenclature part. We learned what nomenclature is, what goes into it. We learned about the committee that oversees the codes, and also how to submit a new strain and to get a validly published name for it. Now let's switch over to taxonomy. As we said, taxonomy is there so we can assign organisms to taxa and also provide taxonomic ranks, right? We had this example here showing that taxonomy means which box represents the species, right? And again, there's no official taxonomy or a taxonomic code, right? But there are common conventions, best practice in the field, and then of course all the papers and manuscripts are peer reviewed, so that kind of reinforces this best practice. But yeah, that's probably a very difficult question, right? Which box represents the species? Let's have a quick look at the species concept here. Now, if you look up the biological species concept, that goes back to Ernst Mayr in 1942, and here he defined a species as a population of organisms that can reproduce with one another and are reproductively isolated from other populations, right? So that works very well, I would say, for animals, probably a lot of plants. But it does not really work for organisms that reproduce asexually, such as prokaryotes, right? Because the definition is they can reproduce with one another. Nobody mates with me. Microbes, prokaryotes don't need a mate. They reproduce asexually. So the question is then, well, okay, where does that leave us? How do we define a prokaryotic species? But the short answer down here, there is no final conclusion and consensus, right? People say that is the perfect concept to define a prokaryotic species. It's still an active area of research, and different ways of going at this topic have been proposed. And that is a screenshot from a paper of Doolittle and Papke in 2006. And they just show a few examples of what you could do, right? You could actually say, okay, we could have an ecotype of species and look at niches that limit the gene flow, right? So there's a niche in between that limits the gene flow, so maybe that is an ecotype species here. Another idea is to go with a concept that's similar to biological species. That means basically you have a species here, and then there is some kind of barrier to recombination, and then that divides all of a sudden into species. Another option is, well, maybe it's just a random process, a random splitting of lineages, right? And actually the species concept doesn't even apply to prokaryotes. Maybe it's completely arbitrary, right? So those were just some hypothetical ideas. Where have we landed then after, I would say decades of discussions? Let's say in the 21st century, what's mostly accepted is a polyphasic species concept. What that means is that you actually use a wide range of techniques, right, to define a species. What are those techniques? I listed three common ones here. First of all, luckily we have a lot of genomic information now. So you can use this and look at the similarity, right, to other genomes. Originally, that was Dna hybridization, then 16S RNA similarity, and now we're working with genomes, so it's mostly average nucleotide identity, ANI, that we can use to actually delineate a species. Now, we should also look into the evolutionary history, because strains should form a monophyletic group, and that is determined by phylogenetic inference, right? Calculating phylogenetic tree. Then the third one, I would say that it's a good addition to have, right? Is the phenotype information. So strains ideally they share a trait that can serve as a diagnostic of the species. That could be a biochemical test, but maybe they can degrade or produce a certain substance. It could be characteristic morphology, right? That additionally enhances the species concept, right? And as I said, there is kind of a community consensus right now, and that is usually enforced by peer reviews, right? Methods are phylogeny and also genome similarity, right? Well, let's look into this a bit more. How did it actually change over the years, right? So, yes, criteria for delineating a species driven by molecular techniques, right? So I exclude phenotype and whatnot, because I see that more as an additional information. Like, yes, well, okay, I start with the phenotype because in the early days, there was nothing else there, right? One had to use morphology, culture conditions, maybe any, pathogenic abilities of the microbes to define it. Now, then in the 60s, Dna hybridization was developed and used widely. And that is a laboratory method. So you really heat up the DNA to certain temperatures and see when it realigns or reanneals with the DNA from a different organism, right? And you can measure that. So it's a lab-based method. Then in the 70s, it all shifted. The 16S became popular originally with Carl Woese, and it was then used widely in the field. And after a few years, the 16S was then the main workhorse, I would say, became like the gold standard, right? To delineate species and also to reclassify higher taxa. In the 2000s, as I mentioned, everything got cheaper, including sequencing and computer power, right? And we could actually then calculate not only single gene trees of the 16S, but concatenated protein trees, right? And those were used also to look into the species concept. In 2005, the average nucleotide identity, ANI, was proposed as a replacement. Actually, it was already there before, but it was proposed as a replacement for the Dna hybridization. And it was started already, but after that, it was definitely favored over 16S similarity whenever genomes were available. Now, as I mentioned, in the 2010s, we recovered, as a field, right, microbial ecology, microgenomics, recovered thousands, hundreds of thousands of new mags from uncultured organisms, and most of them didn't have a 16S, right? You couldn't do a DNA dehybridization because you had no culture, so ANI was really the method that gained more and more traction. And today, yes, we have lots of genomes. Average nucleotide identity, ANI, has become de facto the gold standard, I would say. And in addition, we use concatenated protein trees, right? That has also yielded or ended up in major reclassifications, and also many new taxa have been proposed. Okay, well, let's have a closer look at this ANI. I think many of you have probably used it already. So it has become the gold standard, as I mentioned, to determine genomic similarity of strains. And obviously that means you can use it to delineate species. In a nutshell, right, you compare to genomes, you look for like matched regions, right? And some regions obviously will not match, right? By comparing those, there's one example here. This one has like 9 and 10 bases matched between those genomes of those two organisms, right? That means 9 out of 10, you have a 90% similarity. And you do this for all the regions because the ANI is the average similarity across matched genomic regions. Now, in addition, one term I should mention is AF, that is the alignment fraction, and it's the fraction of a genome with the matched genomic regions, right? And you want to have a certain alignment fraction between genomes, because if that's very low, for example, it would be like 5%. You only compare 5% of two genomes. That would not be very good, right? So I would rather go for something that's like 50 or 60% alignment fraction for your ANI. Okay, that is how ANI in a nutshell is calculated. I should also mention that's an overview that Donovan Parks from the GDP team put together, that it is a single concept, but you can have different definitions if you look into detail, and that is mainly due to how ANI is calculated. It can be done, several tools use BLAST, others use Mummer, and FastIron8 and uses MeshMap, right? So different ways to actually calculate the ANI. The good thing is, though, if you look at ANI values above 90%, and that's what we mostly use for dealing with species, then they are quite consistent. I found this paper in 2017 where they compared like 4 methods, and you can see that they are actually in good correlation, very high correlations of 99. R is like 0.99, right, between the methods. If you deal with ANIs that are lower, right, so as an example of 70% and lower, then there's quite a big difference between the methods. So just keep that in mind. But for our purpose, we go with 90% and above. So those methods are very, very comparable, which is very good for us. Now, how did we establish an ANI cutoff? And you probably know usually 95% is used as a cutoff for the ANI. Well, that comes out of the historical analysis of based on the Dna hybridization. You can see here the cutoff that scientists settled on was 70% Dna hybridization, and that became the gold standard in the 80s. So then that was translated right into ANI. So if we use all of those genome series cultures, we calculate the ANI. Then how would that translate? Turns out that 70% cutoff over here translates to about 95% ANI. So that became then the new gold standard, right? In the 2000s and later, right? But is there more evidence for 95% being a somewhat meaningful standard? There's one analysis that looked into that, how we delineate species with ANI, and you can see here the same species, right? That was based on genomes, 90,000 genomes in blue. Genomes of the same species, there are exceptions, right? They have an ANI that is like higher than 95%. right? So that worked very well. That means this 95% cutoff that we have derived from the DNA hybridization of 70% actually works well for the majority, vast majority of species that are defined today, right? And based on that, the field overall decided that is a good, very practical cutoff, right? We might have to do this. There is more research ongoing in that matter, right? So I'm not saying that's how it stays, but I think for now that is basically gold standard and what is widely used in the field. Okay, that was species, but how do we deal with higher taxa? Or higher taxonomic rank, I should say, from genus upwards. Well, to delineate those higher taxa, there were a few ideas were proposed over the years, right? I'm not going to go into details, but for genera, one idea was to use a percentage of conserved proteins. Another that we could actually also use the ANI, but that actually shows there are quite large overlaps. It's not a very precise cutoff. We could consider phenotypic traits, which I would say is more an addition, not the main criterion. For other higher ranks, the average amino acid identity AAI was proposed, right, and is used by some researchers. Also, 16S thresholds were proposed by a paper by Yarsa. And the issue with that, for example, is that using those thresholds would mean that we have, more than 1,000 bacterial phyla rather than the, 100-ish we have right now. So that would involve quite some changes. Then another idea is to define higher ranks based on important metabolic properties that they share, right? Two examples here. One could argue that Methanobac, right, they are a group of anaerobic organisms that perform methanogenesis, right? So that's basically what defines them. I should say here, Methanobac, right, that is, despite the bacteria in the name, that is actually an archaeal family. Another example is Cyanobacteria, right? Okay, we can say, yes, look, that's a group of organisms that perform photosynthesis, so they should be all combined. Are there issues with that? I would say yes. For example, there are quite a few organisms named methano something, and I would predict that we might find out in the future once we have those in cultures that some of them actually don't use methane or don't produce methane, right? But maybe another alkane, right? So then, I don't know. We have to change the name. I don't know. But another example is Cyanobacteria. They all perform photosynthesis, or do they? We now know they are very basic cyanobacteria that do not perform photosynthesis. And let's look in one case here that illustrates what happens if you really define it based on metabolic traits. So that is Cyanobacteria versus Melanibacteria. And it goes back to a paper Hugenholtz in 1998. And that was widely cited, one of the first one that actually, said, okay, we need some kind of definition of phylum. And here it says that a new phylum is a monophyletic group that is unaffiliated with an existing phylum. And they also proposed back then 16S cutoffs, right? So now using that in 2014, so it also said, okay, Melanibacteria belong into Cyanobacteria, right? You can see them here, that is Cyanobacteria, that is Melanibacteria, because this lineage is robustly associated. That's robustly associated with other cyanobacterial classes here, right? So that's the argument. They are really affiliated together, so they should be in one phylum. Another paper said, well, Melanibacteria should be considered a new phylum based on an 85% cutoff, right? So a fixed cutoff. Again, who is right, who is wrong? It's taxonomic freedom, right? There is no correct answer in that case. Yes, taxonomic opinion is encouraged, but it does present challenges, right? And we can see one of those challenges here. Now, how do we solve those challenges? Well, one idea is to have a rank normalized taxonomy using genomes, and that brings us to the third part of today's webinar. That is a genome-based taxonomy. And here, obviously, we'll use the Genome Taxonomy database, GDDB, and give you an example using Archaea to show how we standardize taxonomy. Now, let me start with this. Why is taxonomy important? And I hope I have convinced you by now that we need it to assign organisms to taxa and provide taxonomic ranks, right? All the way from species up to domain. That is a very useful tool for microbiologists. Now, how is it going so far? I mentioned that until recently, NCBI was the main taxonomic source, and it is a collection of taxonomic opinions, and that has some issues. One of them is that those ranks are not very well defined. For example, if you ask yourself, what is an archaeal or bacterium phylum? Maybe you came across this question. If you just go ahead and Google it, you maybe end up at Wikipedia, and it says the exact definition of a bacterial phylum is debated, and I think that is true to some extent, right? What is a somewhat popular definition that has been adopted by the scientific community is that a bacterial phylum is a monophyletic lineage, right? That's ideally not affiliated with any other lineage and has a 16S cutoff. And 85% were proposed, 75% were proposed, right? That works for some phyla, doesn't work for others. So it's it's one cutoff that people used for a while. But yes, the lack of those definitions and the fact that NCBI taxonomy is a collection of taxonomic opinion has led to a little bit of a chaos, I would argue. And demonstrate this, I pulled out a tweet to look into the problems with the program taxonomy. You can see this this person here tweeted, are there any good recent publications on how messed up the NCBI taxonomy for microbes is? This seems obvious to many, at least from private conversations. And again, it's not the fault of NCBI, right? NCBI just collects taxonomic opinions, but I would agree it is pretty messed up. How can we quantify this? Yes, and there I want to use IKEA as a test case. I guess everybody here is obviously familiar with IKEA, described by Carl Lewis in the 70s. And then that's a picture or 3 of his publications in the 1990s where he proposed 3 domains, right? 3 domains of life on Earth: bacteria, archaea, and eukaryotes. I was about to move on, but then I couldn't really let this stand because that was the opinion at this time, right? I would say we since have moved on. There are several papers that came out in the last decade that show that actually the eukaryotes are not a domain by themselves, but they suggest they have emerged from within the archaea, right? And you might have heard about the Ascoarchaeota, and they played a major role in this new concept that we actually have 2 domains. It's still debated, right? And I have a screenshot. That's a tree from Jari-san, a PhD student in my group. And here we also recovered the 2-domain topology. You can see here, those are all the Ascoarchaeota, right? And within those Ascoarchaeota, there is a lineage, and that is the eukaryotes, right? So all the eukaryotes are actually within the Ascoarchaeota, and in most cases, they are assisted to the Heimdall archaea. So in that case, the eukaryotes, based on this classification, are actually a class within the phylum Ascoarchaeota. And as I said, yes, this is still debated, but it seems that more and more scientists actually look at the evidence, and that is, well, those Ascoarchaeota have like eukaryotic signature genes that supposedly only eukaryotes have, and especially, the phylogenetic evidence that places the eukaryotes within the Ascoarchaeota. So I wouldn't be surprised if the textbooks are shifting as well, and we're going to have 2 domains in the future rather than 3 domains. Okay, but yes, focusing on the archaea, right? On the archaea here. Now, what are the problems in the archaeal taxonomy? First of all, there is a very uneven application of ranks. That might not surprise you, right? There's a paper from 2000, a review in Nature Microbiology, and that highlights that very well. You can see all those lineages here in different colors have been proposed as phyla over the years. You can see already what the big issue is, right? That about half of the diversity, right, has been proposed as a single phylum, the Euryarchaeota. Versus on the other hand, right, here you have like very small lineages, and that's mainly historically. Like originally, I guess you know that from Carl Lewis, you had the Craniarchaeota, right? There was like this group here, and then we had the Euryarchaeota here. What happened is over the years that people discovered many more lineages for the Euryarchaeota and added them to it, right? For the Craniarchaeota, it was different because the Thaumarchaeota, when they were discovered, it was argued that they are so different, there must be a new phylum, right? And that basically kicked off a slicing and dicing of the group, and now many other new lineages have also been proposed as a phylum, right? And it's, I think, the worst, not the worst, but one example is the Asgards, right? You can see there are even more lineages now, but most new lineages were discovered, those tiny little slivers, they were all proposed as new phyla, right? So very uneven, like this little thing is a phylum here, and 50% of the archaea are phylum as well. That is a very uneven application of ranks. On top of that, you can even see that some of the phyla that were proposed, they actually cluster within other phyla. So yes, it is very messy. Other than that, two more issues I want to highlight. The first one is inconsistencies with evolutionary relationships. So if you have ever calculated or inferred a phylogenetic tree and then decorated it with the NCBI taxonomy, you will see that you get a lot of polyphyletic groups. An example is here in red. That's a tree I created based on those were 122 markers, and then I did decorate that with NCBI taxonomy. In red, you can see what NCBI calls Vosa-archaeota. You can see we have some here, we have some there. Which ones are the real Vosa-archaeota? We don't know, right? Nano-archaeota even worse. You have a group here, you have some here, you have some there, right? So are those Nano-archaeota, this one or that one? Which lineage is it, right? It's very hard. It's very hard to make a decision here. And if you want to automate any taxonomic assignments, that is pretty much a nightmare. Okay, on top of that, we also have the third problem we want to highlight here, and number 3 is a widespread incomplete classification. You can see again everything that's here in a black font. Those genomes were assigned to archaea, right? And again, that is because many researchers they submit the genome and say, I think it's basically it's an archaean, and don't classify it further. That goes in the NCBI taxonomy then, and that's not helpful. Obviously, if your new genome falls into that group, right? What can you say? Oh, I have a new archaean, right? Not very helpful. So one other issue to fix is this widespread incomplete classification. Now, to sum it up, the 3 main problems is ranks are applied very uneven, right? And I should say that example was archaea, but it also applies very well to bacteria, right? Second, we have polyphyletic groups. And third, we have widespread incomplete classification. And on top of that, as I mentioned at the beginning, we have no accepted standards to approach or to assign a taxa, right, to higher taxonomic ranks. And that then yields to cases like this one, where one group describes new lineages and say, hey, we found a new lineage, it's a phylum, we call it Geoarchaeota. And then a little bit later, less than a year, other researchers reanalyze that and say, well, actually, what you call a phylum is just it's a deeply rooting lineage in an existing archaeal order, right? Who is right? Again, taxonomic freedom, right? None of those two is wrong. So those are 2 different opinions. Personally, I also reanalyzed that, and I agree with the second paper, right? If you use the latest inference methods, then yes, that actually is a lineages in an existing archaeal order. Okay, so I guess the message here is that there are several issues in the current taxonomy. Is that getting any better? You probably know the answer already. No, it's going to be the opposite, right? So we are still adding many new metagenome assembled genomes, MAGs, to public databases, to taxonomies. So that will end in what some people have feared is going to be an unresolvable chaos, right? If we don't fix those issues I mentioned up there. Now, how can we fix it? Is there a solution? And you will not be surprised that the solution we propose is the genome taxonomy database, right? So let's have a look and dive into it, into the GDP. Here, let's start with the GDP website. So if you're here live right now, or if you're watching this later, I would encourage you to go to the GDP website. You can scan this QR code that should get you right there. You will then go to the start site. It looks similar to this. And in a nutshell, you can see in this release, the latest release is from April 2000. sorry, it's 2022. And there we have about 317,000 genomes that are included in the taxonomy. Most of those genomes are bacteria, right, as expected, 311,000, and a smaller portion, about 6,000, are archaea. Now, but first, what is the idea behind GDP? You can see it's an initiative to establish a phylogenetically consistent, rank normalized. So that's very important. I will talk a bit more what we mean by normalizing the ranks in a taxonomy. And complete, right? Complete, I mean, we want to provide taxonomic information for all the ranks, right? As I showed you earlier, not archaea and then nothing. No, we want to have taxonomic information from species all the way up to domain for all those genomes. And we have that now, right? And that is for bacteria and archaea, right? Both of them. Now, another way to look at this, if you compare the taxonomic frameworks, there's NCBI. And as I mentioned earlier, it aims to reflect the latest taxonomic opinion as expressed via published literature, right? So what researchers submit to NCBI, and yes, it has to be published in some form for NCBI to accept that if you propose a new rank and a new name. GDP taxonomy is in some regards similar because it also aims to reflect the latest taxonomic opinion with quantitative criteria, right, in addition, that taxa are rank normalized, right, and phylogenetically consistent, meaning monophyletic groups. And that is all based on a genome-based phylogeny. And yes, we can do that now because we have a sufficient number of genomes to do this. So, but how is it done at GDP? How do we actually delineate the ranks? Let's have a quick look. There are a little bit more in-depth look, I would say. There are two ways of doing this, right? The species are handled differently than the rest. So let's first look at species. How do we delineate species in GDP? That's an overview provided by Donovan Parks. Not going to go into too much detail, but we use ANI, average nucleotide identity, right? What happens in GDP is we get our genomes from NCBI. Of course, we run our own QC on that, so only so we can filter out poor quality genomes. We then identify the type material, right? I showed you that is really very important for a species assignment and also later on for taxonomic curation. We select representatives, we form species cluster, and then from there we create the genome database. Now, how do we actually form species cluster, right, from ANI? Let's have a look at that. So as I mentioned, it is based on average nucleotide identity, ANI, right? We calculate those for the genomes in the GDP, and then they cluster based on ANI. Now, the first thing we have to do is we have to find out what is the type material, right? In that case, what are the type genomes, the genomes of the type strain. And you can see we identify those, right, based on LPSN, based on what's in the literature. So that helps us. And then from there, we can then apply our ANI cutoff, and that is a 95% ANI, right? Within that radius, that is considered then to be one species. And it works in most cases, right? In some cases, like here, you can see we allow a slightly higher threshold of like, that is 96%. And that is really because in this case, those two species are effectively and validly published, right? So they are part of the code. And we always try to keep the changes to a minimum if possible. And if it's in the code, we try to, keep that species assignment. So that's why we have to be a bit more lenient here. However, right, should we add this? Yes. And that only happens in a very low number of cases, right? So you can see like in this situation, it was like 380 out of like, about 9,000 cases. However, if those genomes are too close, right, as shown here, then if the ANI is higher than 97%, then yeah, the only thing we can do is that is one species then, and this other type then becomes a synonym, right, of the main one. And that happens in a very low number of cases, but it can happen. Okay, so now we have circumscribed our species based on the type strain genomes. What about the other ones that have no type strain, right? What we do here is we cluster them, And we start with the highest to the lowest quality genome. So let's say in those clusters, right, that would be the highest quality genome. So that gets a 95% ANI radius, the same here and here. And then those become at least GDP species representatives. Can we give them names? Well, most of them have no official names because there's no type material, right? But we do provide temporary placeholder names, as shown here. And the idea for that is that even those names are not, overly meaningful, but we can compare, right? So if someone publishes a paper and say, hey, I found the BOG 4-42 species, right? And someone else has a data set and finds the same species, then despite the name being not very meaningful, it allows us to compare and say, hey, in this case, we have the same species, right? So it serves that purpose. Okay, so if we do this, what do we end up with in terms of species clusters? That is the latest Gdp release, release 207. And you can see out here the outer ring that shows us the species clusters. So the ones that have a type strain, that is like about 20%, right? Then there's a low number that have no type strain but still a Latin name. That happens if we have genomes but not necessarily from the type strain, right? And then the majority, you can see here, and this number actually gets larger with each release, they have placeholder names, right? Because there's no genome, there's no type strain defined, right? The placeholder names, you can see there are a few examples here. It could be it's a new species of E. coli in the bacteria, right? Could also be that there's nothing defined and it gets like a placeholder for the binomial genus species name. Okay, but that way we can assign species to all of the GDP genomes. Now, what about ranks above species? Yes, if you look at that, delineating ranks above species. So objective is to resolve the polyphyly, right? That's one of the main issues I showed you, and to also normalize the taxonomic ranks. Now, for this, what we do is we look into, or we base our analysis on concatenated protein trees. And there we go for taxa and monophyletic in those trees. And second, we use a criterion called the relative evolutionary divergence, or RED, and that allows us actually to normalize the ranks. And that is probably, I would say, the heart of the GDP. So let's have a look and see how that actually works conceptually. So we start with a tree, a phylogenetic tree, as I mentioned, based on concatenated protein alignments. And if you've ever inferred a tree, you know that the branch length here, right, they are actually the inferred evolutionary distance, right? That is a tree, those are absolute values. Now, what we do is in GDP, we use those branch lengths and turn them into relative values. And that is what we call the relative evolutionary divergence. The way to do this is that the root gets a, or I should say, the acronym is RED, right? So the root gets a RED value of 0, and all extant taxa, right, on the tips, you get a RED value of 1. And then those nodes in between, those internal nodes, we linearly interpolate the RED value for those nodes. Okay, what does that allow us to do? First of all, it accounts for varying rates of evolution. So if you have a lineage here, right, that has short branches, which is supposedly a slow evolving lineage, right, then the RED value always goes from 0 to 1, right? If you have another lineage that has long branches like this one, and it's supposedly fast evolving, again, RED values go from 0 to 1, right? So you basically scale those to each other, and you can then compare them. So the system accounts for varying rates of evolution. It also allows us an approximation of relative time, right, since we have relative values. And the idea is then that if you look at two families that they have, evolutionary speaking, right, evolved at the same time in the evolutionary history. And that's why we call both of them families, right? So there's this kind of aspect there as well. And based on that, we use the RED as the primary criterion to circumscribe taxonomic ranks above species. One example is here in the dotted lines. Let's say it turns out that the RED value of a family, right, is between 0.63 and let's say 0.72, right? Then we can say, okay, we go through and say, look, all of those actually represent families, right? If we would use absolute branch lengths, it would look quite different. A fixed cutoff, like like for the 16S, would go right through. But this here, you can see the counts for different variant rates of evolution. So that's the core of GDP, how we actually define ranks. Okay, so how is red applied? In a nutshell, the taxa, right from the same rank, let's say a class, it turns out that they fall within a red interval of plus minus 0.1. I will show in a second what I mean, but let's say the red value of a class is a certain amount, and then plus 0.1 and minus 0.1. Those are the boundaries for all the taxa that fall within that rank. Then we also emphasize that taxa, if you decorate the tree, taxa should be placed on stable nodes. So we look for nodes that have a bootstrap value of 90% or higher. And what that guarantees us, it guarantees a stability between releases, and also if you compare different inference methods. And finally, right, that the decision or how we define those taxonomic ranks should reflect existing taxonomic opinion to the extent possible, meaning we want to keep changes to a minimum. Okay, since we know this, let's look at the GDP again, and that is another way of showing the workflow, but I just want to focus on this one. So once we have our species, right, defined based on A and I, then we de-replicate that. We only need one representative per species. That's really to reduce the number of taxa we have, because next we do infer phylogenomic trees. And those are based on single copy marker genes, 120 for the bacteria, and originally 122 for the archaea. We now recently switched that to 53, but multiple sequence alignment concatenated marker genes. And then once we have those trees, that's when the automated and manual curation starts, right? At the beginning, it was a lot of manual curation, and we try to automate it more and more, but the manual part is essential of this process. Okay, how does it look like? For this, I want to go back to our example of archaea. So we calculated the red value, we inferred the tree, we decorated the tree with the NCBI taxonomy, and then we calculated the red values. And what you get is something like this. You can see we have the relative evolution divergence, the red values here, right? And then here we have the different ranks defined by NCBI. And what you would expect is that we have distinct red value for a phylum compared to a class, compared to an order, but it is pretty much all over the shop, and they're overlapping quite a bit, right? On top of that, the red ones, those actually indicate you have polyphyletic groups. So what we have to do here, we have to resolve polyphyletic groups, and we also have to correct or update the taxonomic ranks, right? For example, here you can see the urea archaea, right? They have the lowest red value of all of them. So that might mean that that's not a phylum, right? That's been broken up in multiple lineages, something we saw already at the tree earlier, right? And on the other hand, here that is Mars archaea, right? Has been proposed of a phylum. The red value would not indicate that, right? So now we can see if we can find another internal node in the tree, or other than that, we have to assign this lineage to another taxonomic ranks. And usually in the beginning, it was a very iterative process, right? You basically look at those ranks, you reassign it when necessary, you run red value again and see how it changes. So after many rounds, then we came up with the GDP version of that, and that is the release R89, right? And you can see we ended up with something like this for the archaea. And here, after we implemented the rank normalization, split up polyphyletic group, corrected rank assignments, you can see what we have here is we have distinct taxonomic ranks for, distinct relative evolution or divergence intervals, red intervals for each rank, right? You have phylum, class, order, family, and genus. You can see there are a few small overlaps here, like in the order. And we really tried to accommodate, so those are again validly published orders, right? So we made a few exceptions there, despite the red value indicating that it might be, a family. But except for those few, all of them follow the red value very well. And you can see then that the Mars archaeota we had here, right, now after several iterations, they were assigned now to an order, which is the order Mars archaeolis. So with this approach, using relative evolution or divergence, using the red value, we can actually fix this problem of uneven application of ranks and have a system, right? So if you, sorry, if you now would use that system, and your tree would look like this, you calculate the red value, and you have a new lineage that has a red value, right? It's tree specific in your tree. Let's say there would be 0.2, right? Then oh, what I have is a new phylum. Like say my red value is 0.75 in my tree, then oh, that would be actually a new family, right? So it really helps to assign the ranks. Okay, problem number one, I would say is mostly solved. The next one, what we have, inconsistencies with evolutionary relationships, right? And that is really where a lot of hours of manual curation went into, because the only way to resolve this is to actually open the tree. We work with ARP a lot. And then what we have to do is first we have to find the type, right? The type material. And in that case, we have here Candidatus enigmachium subterraneum, right? That is where we have it here, yeah, down there in green. So we know the type material is in here. So that is actually the real enigma here, right? Another example is Nanoarchaeum I mentioned earlier. That is in blue, and you can see based on NCBI taxonomy, Nanoarchaeum is all over the place. Now the type is Nanoarchaeum equitans, right? And we have a genome sequence. We can say yes, the type is in here, right? So that is actually the real group, right? And in that case, it was a phylum, so that is the phylum Nanoarchaeota, right? All those other Nanoarchaeum assignments or Nanoarchaeota assignments in NCBI, they are incorrect then, right? And some of them, sometimes they fall in others. For example, this one is part of the Vos archaeolis, right? Others like those ones, they have, there's no type material in that group. So then we actually go and assign a placeholder name. Another, well, I should highlight an issue here is that if there is no type material, and there are still a few cases where people propose higher taxonomic ranks, let's say a phylum, and do not define type material, right? That is a big problem, because then it's really hard to, almost impossible to find out where is the type material, and if this lineage is polyphyletic, we don't know which is the true lineage and which one is not, right? So type material is essential if you propose any new ranks or any new names. And yes, and the third problem, the widespread incomplete classification. As I mentioned, if there is no type here, then we assign placeholder names, right? And that allows us then at the end to have text strings that have all the ranks from species all the way up to domain. So we have no incomplete classifications in GDP. Okay, so implementing all of those rules did cause a few changes, although our idea is to keep changes to a minimum, as I mentioned. What did change? So you can see here in the archaea, right, in the archaeal genomes, if you look at it on a per genome basis, then 93% required changes to the existing taxonomy. That sounds like a lot, but that is mostly because the classification was incomplete, right? If you break it up by rank, you can see active changes in red. That was when we actually went in there and we had to actively change the taxonomy. Passive was, that's when there was no information, we filled it in, right? And in light blue, those are unchanged. So you can see that overall, we only changed about 10%, right? 10% of the taxonomic assignments for each of those ranks, except at the phylum level, right? The phylum level, we had quite big changes, about 70%. And the reason for that, I think you can already figure it out, is mostly because, well, the uneven application of ranks, especially phyla, right? So to show you that, again, that's the R89 taxonomy, where we compare GDP versus NCBI. So that is a protein tree based on 122 markers, and I decorated that with the NCBI taxonomy in orange and the GDP taxonomy, that the new derived one, in blue back then. So you can see in orange, that is, I should mention, yes, that is only one representative per species, right? And if you do it that way, you can see that almost like 2/3 of all the archaea, according to NCBI, belong to a single phylum, the Euryarchaeota, right? So we had to split this up according to the relative evolution of divergence, the red values. And in this case, we end up with 3 main phyla: Halobacteriota, Thermoplasmadota, and Methanobacteriota. Also, the 2 little ones here, the hard and hydrothermarchaeota, right? On the other side, it's over here. On the other side, here that is what was known as the TAC superphylum, right? That is Thamarchaeota, Eikarchaeota, Crenarchaeota, Corarchaeota. And here that was originally at some point called Crenarchaeota, then it was split up in multiple phyla, then it was combined on the term TAC. Anyway, the whole TAC superphylum, based on the red value, is assigned the rank of a phylum, right? So the whole thing is a phylum. And for this one, we use the name that was proposed earlier, Thermoproteota. Also, the Asgards, right? All the Asgardarchaeota, well, all the Asgards got the rank of one phylum. Asgardarchaeota is seen here. And then here we have what's also known as the Deepan supergroup. And there are many small, deep diverging phyla, right? And I think that is probably everything here is very stable between releases, right? In the Deepan, since we have a low representation of genomes and long branches, that's maybe where we're going to see a few changes in the future. The rest seems to be very stable. Okay, so that is how we initially started the GDP taxonomy database based on here as an example. How we're doing right now, that is already outdated. I should update this slide, but last year we had over about 30,000 users, right? And about 300,000 page views. So I would say the scientific community has adopted GDP very well. There are several papers here. Again, I don't list all of them. And we also have developed GDP TK, which is a toolkit that allows you to classify your own genomes. And I'm going to talk about this a bit later. So yes, very positive feedback from the scientific community, and we're very happy about that, right? Of course, there were also critics, right? There was criticism. And when we submitted our paper, the archaeal GDP paper, one of the reviewers actually summarized the expected criticism very well. What this reviewer predicted is exactly what happened. And I want to share it with you here. So this reviewer said, I predict that this newly proposed system, GDP, will lead to resistance from 3 kinds of researchers, right? Number 1, traditionalists, shown here, who want to keep living like in the old days and who keep ignoring the wealth of microbial diversity that has been overwhelming us since the dawn of massive parallel gene sequencing. These researchers keep refusing DNA as type material with the sole argument, we haven't done this in the past. Yes, and I do have encountered those individuals, and there's not much we can do about it. I might suggest that time will solve this problem, right? And young scientists know exactly how to work with DNA, know the value of DNA sequences, and this opinion might go away in a few years. Okay, second researcher, and yes, there are definitely many of those out there, people who named one of the taxonomic groups that are now being renamed. I don't like the term renamed, reclassified maybe. And I've seen this in conferences in which the authors presented their study, GDP authors. People fight for the names they came up with, often without any scientific justification, but simply because they are emotionally attached to the name, right? And yes, that's definitely the case. Someone named the microbe and then named the microbe, proposes a phylum, and then GDP comes back and says, well, actually, that is not a phylum. And it's a class, and according to the taxonomic rules, the name for the class has to be derived from an order, or from a genus, sorry. And that way, that means that the name has to be updated. People are very unhappy about that. Luckily, many researchers that have this opinion have come around in the recent years and have realized that yes, we might lose a name, but it is an advance for the greater good because we have a rank normalized system. Okay, and then the third kind of critics were scientists who have serious concerns about the validity, right, of rank normalization about our system that I showed you. And as said above, as far as I understand, this was done scientifically in a scientifically just way. I would say it has been done. And yes, in this review, then also mentioned that personally, I'm a pragmatist, but I might not like that some of the groups being renamed. I see the greater good that comes out of this newly rank normalized system, right? So there are some changes in the long run, but I think we're way better off there are some changes in the short term, I want to say, but we're way better off in the long run. Okay, so yes, what can we do about this, or how can we respond to that? We can definitely respond to number 3, right? Those are scientists that have serious concern about our rank normalization system. And one argument that came up in the discussion is, well, it's too early to really work on the archaeal taxonomy because we don't know where the root is, right? Without knowing the root, we cannot establish the ranks. And we tested that, and our argument is, yes, we can establish the ranks, because for the red value calculation, it matters very little where the root is. So if you look at this here, that is a standard GDP way to calculate the relative evolutionary divergence, the red value. And the way it works is to calculate the red, we look at the main phyla, right, that have at least two daughters, so 2 classes, and then we root the tree on each of those phyla, and then we calculate the red value, and the final red value is the median value of all those calculated red values. So basically, it accounts for different rooting scenarios. That is very good, right? I think that's a very, in our opinion, stable way of doing it. But what if it turns out that, if at some point as scientific community agree on where the archaeal root should be placed? So we tested that. There are many papers that suggest the root should be actually here, right? The root should be here at the DPAN superfile, right? If you do this, and then we force the red value to be calculated from only a single root, right? You can see that the red value changes very little. You still have the same intervals. The same if we go for a root that has been suggested within the ureaciota as defined by NCBI, and that those also suggested to put the root in the middle of the eukaryotes. If we do this, ureachia, sorry, the middle of the ureachia. If we do this, then force the red value to be calculated only of the single root. You can also see that the red intervals move like very little. So based on that, we argue that we don't really need to know where the root is. We can still very well calculate the red value and come up with a normalized, rank normalized taxonomy, right? And if in the future we would decide where the root is, we can easily switch to one of those scenarios if the scientific community agrees, right? Which is probably hard to achieve. The other question is, okay, well, what if your system is based on a tree? So what if you use different markers to calculate the tree? If you use different alignments or different inference methods? So we also tested that. And before I go into this, I should show you this, the color code that I'm using in the next slide. So when we created the trees, then what we're looking for is taxonomic stability, right? So we want to see which group that we gave a taxonomic label is actually monophyletic, and then we'll get a green color. Or the other case, if the group turns out to be polyphyletic, let's say another group jumps into it, right? Then this group would be colored in red. Now there are also cases where it stays monophyletic, but a single genome or a very small amount of genomes jumps in, right? And for this, we actually calculate the F measure. And if the F measure is greater than 0.95, then we call this operational monophyletic, right? So there's one case where we have 24 taxa in here, and then an additional one jumps in here, right? So it's still mostly monophyletic with this one exception, and that's why we call it operational monophyletic. So basic monophyletic is what we want. Operational monophyletic is what we can live with, and polyphyletic is what we want to avoid. If we do this, right, you can see here, we tested this for the ranks of phylum, class, and order, and we really included a range of phylogenetic trees based on different markers. So usually we use 122 markers, right? We also looked into ribosomal proteins. So that is RB1 is 16, RB2 is 23 ribosomal proteins. We also compared it to the SSU, the 16S trees, and we compared it to 53 markers that we used. We employed different methods, right? IQ tree, XML, phylobase, and compared between those trees. And we also looked into trimming, right? Because it depends how you trim your alignment. And we had different ways to trim the alignment, to reduce compositional bias, to remove fast evolving sites. We even included a super tree approach, which is here the Astol tree, right? We even recoded in the 4 character states. So we performed all of this, and you can see the take-home message is overall, right? The taxonomy is very consistent, right? It's mostly monophyletic or operational monophyletic in green and in yellow here. There are a few cases where you have a red color polyphyletic. You can see the worst case is the 16S phylogeny, and I think that is not surprising because for this, what happens is, right, not all of the genomes have a 16S. So some of them have shorter ones, which we had to exclude. So they're quite less taxa here, plus it's only a single genes, right? So I would actually trust this one the least, and you can see quite a few polyphyletic groups here, which is exactly what expected, right? The other one, I guess, that has more polyphyletic groups is the Astral tree, and that not that many actually. Some at phylum, and then almost nothing here at class and order. But that is a super tree approach, which is quite different than our concatenated alignment approach. But overall, I would say there is a very high consistency in these comparisons of the GDP taxonomy, regardless what kind of approach you use to actually calculate the phylogenetic tree. Okay, another question that came up is, well, you have shown that the data set, the genome data set, especially MAX, is changing so quickly, right? There's so many new genomes becoming available, showing up in public databases. So can a GDP actually deal with this large increase? Wouldn't that cause a shift in taxonomy? And we tested that. And by we, I should say, Pierre did actually. So Pierre tested it. He simulated the dataset expansion and said, okay, all the GDP taxa we have right now, or I should say all the genomes we have right now, how many of those were present in 2017, 2016, 2015, right? You can see he did this in 2018. It's quite a lower number, right? So as we know, the number of genomes increased quite a bit over the years, and that's archaeal only. So what happens if we infer the tree with only 500 genomes or 360 genomes, and then apply the red approach, right? What do we see? And we did exactly that, and you can see that again, it is a very, very stable, right? In most cases, we have all the groups are monophyletic or operational monophyletic. And that had mainly to do with the fact that the red is calculated on the main phyla, as I mentioned, right, that have like two daughters, 2 classes, and then rooted on each of them, and that's how we calculate the red value. So that is from our side, I think that's a very reassuring result, and it also means if the genome numbers keep increasing the way they are, and they might as well, do that, then the GDP is well set up to actually deal with those, and we won't see any changes in taxonomy that are caused by increased number of genomes. Okay, yeah, one more slide I have, that is just a test case I want to throw in there. Again, from a paper that Chari did, and in this case, she looked at all the new lineages were proposed for the Ascodarchaeota. And you can see most of them in brackets, they were proposed, most of them proposed as phyla, right? Almost all of them. And then we used GDP and the rank normalization to deal with this whole group. And as I mentioned, the whole Ascards come out as a phylum, the Ascodarchaeota. And then in there, you have like different lineages, right? Most of the phyla, proposed phyla, published phyla, right, are actually come out as the class, right? Some of them are actually even below that, like Odinarchaealis was in order, right? Or yeah, no, the other ones were also classes in that comparison. So that shows that you can actually use the GDP to resolve some of those groups that are heavily debated, and we have rank normalized assignments, and hopefully researchers will adapt that in the future. Okay, GDP releases. Yes, I mentioned that already earlier, that GDP is released every 7, 9 to 12 months. So we try to be as quick as possible, but it is, as I mentioned, a lot of manual curation involved in that. The latest release is from April 2002, and I can tell you we're already working on the next release. Again, GDP website. What I want to point out, you might have explored the GDP website by now already. 2 features I want to highlight, I think that are very important, that are, that I wouldn't say they're hidden, they're there, but it's good to know about them in the first place, right? The first one is the taxon history, something that we also use a lot. And you can find this here under browsers and then taxon history. And what it shows you, you can actually choose what you want to compare. It actually shows you what happened between the different releases, right, of GDP, what changes there are. And then it also shows you how that compares to the NCBI taxonomy. For example, here I have the Archaeota, right? And you can see that was the latest GDP release, right? We always get new ones in here. So not present means that the new ones, and then the new ones get assigned, right, to different lineages. So let's say the current release is 207, and we have all those different groups compared to NCBI. So 3,000 genomes in NCBI are assigned to Archaeota, but in GDP, based on our phylogeny, our rank normalization, our removal of polyphasic groups, we can actually really resolve and show what they really are, right? They're assigned to a wide variety of groups, right? And that was fairly consistent between this release and the last release. You always get new ones each release, they get assigned to, their representative taxon. So that is very helpful. Excuse me. That is very helpful to see what happens between releases and also to compare GDP and NCBI taxonomy. Sorry for that. One more thing about the website. You can download the data here. And what we have is we have taxonomy files and the trees for bacteria and archaea that you can obtain. And that is under go to downloads and then release the latest. And that's always where the latest releases. I'm not going to go into detail, but if you want to know information about the files you find there, I would say go to this folder that describes each data file here. Then we also have a description of the methods here, right? And this file here, that's probably one that you want the most, that has more than 100 metadata fields for the genomes that are in GDP. That includes, completeness, contamination, GC content, genome size, NCBI taxonomy, and so forth. So that is probably, a very useful file. And then the other one is taxonomy file. So that has basically the ID for each genome and then the full text string associated with it. Yes. Oh, one more thing about the webpage. If you want to contact us, right, the best way is actually use the Gdp Forum, and they just got a new look recently. And you can see here, depending if you want to talk about bacterial taxonomy, archaeal taxonomy, general GDP, release notes, or the GDP toolkit, right? You have different subforms here you can post, and that is the preferred way to get in contact with us. And we are usually pretty good replying to any questions that are posted in there. Okay, then I guess, yes, before we end, I want to have a look at this one here, the GDP TK, the toolkit. So there is, it's a publication associated with it. There's even a new one coming out right now for the second version. But basically what this toolkit allows you is to assign objective, I would argue, taxonomic classification to bacterial and archaeal genomes. It uses the GDP rank normalized taxonomy, and you can get it from GitHub. Now, if you're working on the bioinformatics workflow, where would GDP TK fit in? Basically, after you have your quality check next, right? I think you've seen in the past. We get the reads, QC, assemble, bin them, and then run QC with checkm. And there's also a new version, checkm 2, coming out to assign the quality of max. And then whatever passes a certain quality threshold, right? In GDP, that is actually defined as completeness minus 5 times contamination, has to be above 50. But you can set your own threshold, obviously, right? Feed those in a GDP DK. I would not feed in like really crappy max. If they're 10% complete, I think we have a warning flag now in there. That would not help, but I would go with like high quality max here. Feed them in GDP DK, and you get then a taxonomic classification, right? What we always say, that gives you an idea of the classification. If you really want to go deeper into that, for example, propose new names, whatnot, I would always encourage you, right, to calculate the de novo tree, right? And you can then also employ a rank normalization on that tree, and that based on that de novo tree, you actually propose a new lineage. I would not do it based on GDP alone. That's really more to give you like a first glimpse of the taxonomy. Yeah, one more slide. So if you have questions about GDP, then there's the forum, and especially Pierre. He's the developer behind GDP together with Donovan. And in a nutshell, right, just leave this up for reference here. The whole workflow, the classify workflow, can be called by classify _ workflow. And then what you have to point it to is the directory with all the genomes in there, right, all the bins. Usually those are FASTA files, FNA files here, and then an output directory. Yeah, for the computational demands, it was at some point 150 gigs of RAM that you use, right? That has in the meantime increased to almost like 300 gigs of RAM to run, and to actually solve that, because that's a lot of RAM to have, right? We are working on, or we have already released, you can get on GitHub, the next version, GDP version 2. It's on bioRxiv right now if you want to check out the paper. And that actually has a different approach, a divide and conquer approach. And so user genomes are initially placed into bacterial reference trees only at the family level, right? So it's way faster because it only goes down the family level, and family level representatives, I should say. And then this is followed by a placement in an appropriate subtree, a class level subtree, right? So this divide and conquer approach makes it faster. I don't have the in this paper you can see a comparison in terms of speed. It makes it faster, and mainly it reduces the memory requirements, right? Makes it easier to run. Okay, I should also say there are other classification tools out there as well. And Donovan Parks did this comparison in 21. And for this one, he used the genome. That was, you can see there's the genome ID that was classified as Bacteroidealis bacterium at NCBI, right? It was a high quality one. It had a 16S. It had less than 1% contamination. So by all accounts, a good quality genome. Running that through GDP, you will get a full text string, right? Starting with Phylum Bacteroidota all the way to family, genus, and species. You can see those are then placeholder names, right? Because there are no official names available. Another tool is MEMAG. You can run that uses AAI and ANI. And in this test case, that would classify to Bacteroidota, possibly, or the Bacterialis, and then stops there, right? Another one is TYGS, type strain genome server, and that came up with an answer saying it's Bacteroidealis xylani solvens, with a warning indicating that the assignment might not be reliable, right? So that should just tell you, A, you have different sources out there, and B, I'm very happy with this outcome, because I would say in this case, GDP probably gave you the most usable result, right? If you're not into running GDP, it runs the command line. It has been incorporated in other environments. One of them is Kpace, right? And that can be run, that has a GUI, so you don't need any command line knowledge to actually run that. Okay, with that, let's come to our conclusion. Nomenclature, we have learned it is well defined, has rules for assigning and validating names. Then taxonomy, right, is different. It's the assignment of organisms or strains to taxa and taxonomic ranks. and there is no taxonomic opinion. There's no official taxonomy, but plenty of opinion, right? And species delineation, we looked into that. The polyphasic approach is usually applied, and when you have genomes, A and I became the gold standard. And then introduced to GDP, a scalable genome-based taxonomy providing classification to the uncultured majority, all based on genome sequences. Okay, with that, yes, one more slide before we're done. So earlier we talked about naming a species, right? That you have to have type strains, you send to a culture collection. It's all very nice. But what if you have uncultured species? If you have, for example, a MAG, right? Genome sequences, then you cannot do this, right? And that became a big problem because, as I showed you for the species cluster, the vast majority now are uncultured, right? We'll get more and more uncultured species, and that kind of gave rise to the C code. Now, there was a publication earlier that genome sequences should be able to serve as a type material, right? And then the community got together in 2020. This roadmap was published for naming uncultured archaea and bacteria, right? Then this was brought to the ICSP, right? The committee that oversees the code, and there was a proposal to accept DNA as type material. Now, unfortunately or fortunately, this proposal has been rejected, right? Meaning that the codes, the ICNP will not include, will not accept DNA as type material, and that has then led to a, I don't know, a hard fork, if you will, in 21. Efforts started to produce a new code, the C code, right, that allows DNA as type material. And if you want to check it out, I would say follow the URL here, or you can also follow C code on Twitter. And yes, that is coming along nicely. And that will also mean that pretty soon we're going to have two codes, right? So C code is going to take over most of the assignments of the code, right? The Procreate code. But at some point, there might be, well, there will be DNA-based type material in C code that is not in the ICNP, right? So we'll see how that goes, but I think most scientists are on board and saying we need the C code to address the nomenclature, right? The taxonomy is addressed by GDP, but we have to address the nomenclature of this high number of new MAGs. Until this is done, if you name a new lineages and you have MAG and SAGs as type material, right, then I would point you to this paper by Maria Chewotchina that gives you a rough guideline how to go about this, you know, what quality thresholds you should use, how do you propose a name. I think that's very helpful. And with that, I want to finish, and I think my last take-home message here is, if you name a taxon, right, let's say an archaeal phylum once, right, what do you do? Don't call it Bob and walk away, right? That is terrible. That creates a lot of headache for all of us. If you do this, please, right, define a type, define a type genome, define a type species. In that case, it will be Bobarchaeum Bobby, right? Then you have the genus name Bobarchaeum, then propagate it up the rank to Bobarchaeaceae phylum, Bobarchaealis order, Bobarchaea class. And if you believe it's a phylum, then you know it's called Bobarchaea order, and that is very important because that really helps us in the taxonomy. So please always designate type material. There is a slide of all the resources. Maybe we can post it below the link to this YouTube video for nomenclature, taxonomic, and classification tools. And with that, I just want to point out the GDP team. If you see us on a conference, feel free to approach us, right? We're happy to talk to you. And here I want to say thank you to the GDP team, to all collaborators, to our funding sources. I want to say thank you for your attention, and I'll take any questions you might have. Also, if there are no questions right now, then I would encourage you can send me an email, or even better, you could go to the GDP website and to the Gdp Forum and ask your questions there. Okay, thank you all.
Hey, Christian.
Yeah.
There are 4 questions in the Q&A.
Okay, perfect. I will have a look at that. Yep. Okay. Thank you. Yes, the first question, a good one. How are uncultured species named? Yes, And I think I have answered this question already. That is addressed with the last two slides we had. We will eventually be able to use the C code for that, right? For now, you could follow this paper that I pointed out, where we described how you can use high quality draft genomes to name your lineage, right? It would be still based on the current system. You would still put a candidatus in front of it, right? And it will be out there in the literature. We can also incorporate it in GDP, but it will not be accepted by the code, right? Because the code only includes cultured species. So that's what you can currently do, right? Follow that procedure. But the C code, and everybody's working on that feverishly, and hopefully there will be a website available soon, and you can then go there and enter your proposed name, and the C code will then check if this name works with the rules of the C code and give you then a feedback, right? I don't have the details at this point, but you also at some point need to provide a link to submit a publication, the new name. But the idea is that then C code will validate the name even before it's published, right? And that way you have it included in the C code, and that's the way to go about publishing names of uncultured species in the future. Okay, another question. Can you discuss the GDP red scores and how they're used to define new species and genus? Yeah, I think I went over this too, right? As I mentioned, the way they are calculated, we then have like intervals, right? Red intervals, they are specific for each tree, right? So if you calculate your own tree, they don't have to look the same as they look in a tree that I showed. But usually what you have is you have specific intervals for different ranks, one for, say, phylum, another one for class, another one for order, right? With like very little overlap. And that then allows you to see, to place a new lineage, to assign a rank to your new lineage then. Another question, are type species genomes required to be culturable, submitted to ADC DSM-set? Yeah, so there are type species and type genomes, right? If you go with a, well, let's put it this way: if you have a culture, right, then you would go the traditional way, submit it to 2 culture collections, and then publish it in IGSM, and then it will be eventually validated by the code, right? That's one way to go. It has to be in a culture. There are certain cases where we have microbes that cannot be grown, in an extended culture by themselves. So they, for example, need a partner. And that's a little bit debated right now what to do about those. There are a few cases that they could still make it and could still be verified by the codes, but those are a little bit more difficult cases. If you have only a genome, right, and there's no culture associated with it, yes, then you would go in the future, you would use the C code, right, the alternative to the code, and that way you get your name validated and have a new name for your lineage that you propose. Hope that answers your question. And the last one, how to make a rank normalized tree to determine a new rank. Yes, in that case, I didn't go into detail into that, but you can basically use any tree of your choice, but we usually use ribosomal marker proteins to infer a tree from that, right? You could use any other tree as well. And after you do that, there's a tool called PhyloRank that is also available on GitHub, and you can feed PhyloRank a tree and a taxonomy file, and it will then decorate your tree, for example, with the GDP taxonomy, right? And that way you get a feel for the taxonomy. It can also calculate the red value for you, and using that together, you can then basically establish the new taxonomic rank for your new group, right? And I would, as I mentioned, I would do this in addition to GDP-DK, right? So GDP-DK gives you an initial result, and we do our best to make this as reliable as possible. But if you propose a new lineage with a new name, I would also always encourage you to infer your own tree, use the PhyloRank tool to calculate the red values, to decorate the tree, and then make an educated decision of what rank you're going to assign to this new lineage. Yep, thank you. I think those were all the questions we have for now.
They are. Thank you so much.
Yeah, no worries. Thanks for having me. And as I said, more questions, please go to Gdp Forum and we're happy to answer them there.
Well, thank you for the presentation and have a good night.
Well, thanks, Heather. You too. See you.